Good afternoon, ladies and gentlemen. My name is Aneta Křížová, and it's my great pleasure to welcome you to our webinar about quantitative phase imaging for live cell analysis. In our webinar, first, the principle of QPI will be explained, and then we will look closely on the data that can be extracted from quantitative phase imaging. Next, we will introduce to you our Telight patented solution for QPI and our analytical software. Finally, we will show you some possible applications of QPI in cell analysis with special focus on mechanobiology studies. At the end, we will look at your questions in the chat and discuss them. The webinar should take about 30 minutes with additional 10 minutes reserved for the discussion based on your questions. Please note that you can ask questions during the presentation by typing them on the chat. And now I would like to pass the word to my colleague, Dr. Zuzana Nováková, the main speaker today.
Thank you, Aneta, for a nice introduction. Also, good afternoon to everybody who joined our webinar. So let's just jump in. This webinar will be about quantitative phase imaging, but first of all, what are the troubles when you want to visualize cells? So the trouble here is that the cells are actually phase objects, meaning that they affect the phase of the transmitted light, the light that passes through the cells, and not the amplitude of the light. Therefore, they appear transparent, and they are not detectable by human eye, and also difficult to detect it for the cameras. Here's a typical bright field image from the microscopes. There are some methods how you can enhance the contrast of such transparent objects. Some of them are label-free, typically phase contrast, different interference contrast, high-pass filter. These techniques are good for live cell imaging, but they are not quantitative. They allow you to visualize the cells, but not in the quantitative way. Then there are techniques based on the labels, typically fluorescent microscopy, which allows you to visualize cells quantitatively and specifically. You can label specific structures, but there is always a question of the fluorescent labels interfering with the cell structures, causing toxicity, phototoxicity induced by the light, or photobleaching of the labels themselves. And then there's quantitative phase imaging, which we will be talking about today. Quantitative phase imaging uses a nice principle of splitting the light into two beams. So in the microscope, there are two arms. One is called the object arm. The second one is called the reference arm. And what happens is then the object arm, usually cells, another sample, is placed. And in the reference arm, only the reference sample is placed. And you can see that the phase of the transmitted light is affected by the cells and not by the reference sample. At the end, these two beams are brought together and they interfere at the camera plane. So at the camera, we detect the hologram, the interference pattern of these two beams. Here is an example of such a data. So on the left, you can see the hologram, and on the right, the phase image that is reconstructed from the hologram. Phase image, you can see cells with nice contrast. And in the hologram, you can see that the raw data here in this technology are really the interference patterns, interference of the two beams. And the hologram stores the complete information about the transmitted light, about its phase and amplitude. This allows us to reconstruct precisely the phase image. But what's more, the phase image is quantitative. It means that each pixel of such an image carries the information about radiance. How much was the light delayed compared to the reference beam when the light passed through the cells? So, quantitative phase imaging allows to visualize cells in the quantitative way. So at the end, you get the image that is in radiance. But you can take one step further and directly quantify the cell dry mass, because the phase shift, how much is the light delayed when passing through the cells, is actually depending on the cell dry mass. So higher the cell dry mass, higher the phase shift. So quantitative phase imaging allows you not only visualize cells in the quantitative way, but also to quantify the cell dry mass. And that's not the only advantage of this technology. Compared to other label-free techniques of cell visualization, quantitative phase image allows precise background discrimination, and therefore it's a suitable technique for the cell segmentation. You can see here how quantitative phase imaging data can be easily segmented because of the precise detection of cell outline and the precise method sensitivity. Therefore, these data represent a suitable source for the advanced image analysis. So there are already several papers that use quantitative phase imaging data for machine learning. So what data can you actually get when you use this technology for looking at your cells? I already mentioned that you can directly quantify the cell dry mass. When you segment the objects, then you can assign many morphological parameters and describe each object by its area, density, circularity, and many more. If you perform time-lapse experiments and then segment the objects, you can also track the objects in time and describe their movement by, for example, speed, motility, meandering index, and many more tracking parameters. If you combine quantitative phase imaging with fluorescent imaging, then of course you can combine these two data together. This is the first part of the webinar when I wanted to explain you the principle of quantitative phase imaging and what data can be derived from such images. So to summarize the main advantages of this technology, first of all, it's label-free technology. It allows to visualize cells without the need of using any fluorescent labels. It's quantitative. It allows to precisely quantify the cell dry mass. It's of low phototoxicity because it doesn't depend on the intensity of the light. It's based on the interference of two beams. There is no photobleaching, so this technique is suitable for performing long-term observations. It's a single-shot imaging technique, meaning that the acquisition speed depends only on the speed of your camera or on the software. It provides high-quality data of high contrast, which is suitable for cell segmentation and tracking of the objects. And it's highly compatible with the artificial intelligence-based image analysis. So this is about the technology. And what do we do here at Telight? We designed and produced Microscope called QPhase, which is based on this principle. So you can see that in the beginning there is a light source, and then the beam from the light source is split into two arms. So you can see that on the image there are two condensers, two sets of objectives, so two identical optical pathways for the light beams. On the right side, in the object arm, we have equipped Microscope with fully motorized stage and stage drop incubator to ensure the right conditions for the cells in long-term experiments. So this is a diagram how these two beams pass through the microscope and they interfere with the hologram. And from the side, we decided to couple the microscope with the fluorescent module so we can combine QPI and fluorescent imaging. And there is a reason why we do that, because label-free wide-field methods as phase contrast or differential interference contrast or QPI, they are all label-free with low phototoxicity, but all of them lack the specificity. So when you want to visualize specific structures, you have to stain them. That's where the fluorescent steps in. And this is also the reason why we combine these two imaging modalities on our microscope. The data can be, of course, easily overlapped in our software. And also I would like to mention that the microscope is equipped with different magnification objectives. So the user can decide if he wants to study larger cell population in bigger field of view or focused on the details in cells. And the microscope itself wouldn't be complete without our software. The software is called SophiQ, and in the live mode, it controls the microscope and allows the user to set up acquisition, which can be multimodal, multi-position, due to motorized stage. And in the offline mode, it's quite a powerful analytical tool, not only for viewing the data and directly exporting them to videos or images, but also in the software, automated cell segmentation is performed, and then the data analysis is conducted. So in the software, you can browse the history of every cell. The software is quite interactive and allows also to perform population gating, which means that you can use the dot plots and select any two parameters that you are interested, and then filter out the cells that have some specific parameters. So you can gauge the subpopulation in your data. Here is the short video, just a screen capture from our software when I wanted to show the sensitivity of the technology, so you can see that the background is really flat and the signal is coming from the cells. Also, each cell gets a unique ID number, so you can track the object in time. You can display the data for each cell in the tables in form of time graphs and histograms. Here's an example of histograms for the mass parameters. These are the dot plots that I was talking about, and the modulated rows diagram for describing the modulated mass. So now I'm choosing two parameters and the dot plot for them, and this is the population gating. So you can use different types of gating, and then you can see the color-coded cells in images, in the tables, in all the graphs. So the system has quite nice colorful connectivity, and then you can play the time lapse and export the graphs directly in the format of vector or raster graphics, or in videos, or in CSV to perform the statistical analysis in different software. So this is the SophiQ software, but to learn more, that would be another webinar. Let's go back to the research applications of the quantitative phase imaging in general. So this technology is not reserved for any for a certain cell type. You can study cancer cells, stem cells, immune cells. You can use it for studying the effect of the drugs in pharmacology and mechanobiology, which will be the spotlight at the end. And since it allows direct characterization of the cell dry mass, then it's really suitable for studying processes where the cell dry mass is indicative. For example, cell proliferation, cell growth, cytotoxicity, these subtle changes of cell dry mass. Even it looks that on the images the cell is not dying, but there is some toxic effect already happening. Studying cell cycle, different waves of cell death. I will show you examples later. So let's see some application teaser. Due to high phase detection sensitivity in our system, we can quantify even the minimal changes in the cell dry mass and to monitor cell cycle really precisely in more detail with a label-free approach. And there are two videos that I want to show you of symmetrical and asymmetrical cell division. So you can see that this one cell that we are monitoring is gaining mass, typically the S phase of the cell cycle, then it divides. So you can see the drop in the cell dry mass and also the drop in the cell area, that's the lower graph. And after the division, the new cell is tracked, and you can see that these two cells have similar cell dry mass and cell area, so typically for symmetrical cell division. In case of asymmetrical cell division, this is an example here. Again, you can see how cell is gaining its mass. It also increases in the area. And then the mitosis starts, so the cell shrinks. Nuclei is also divided, and then we have daughter cell, and the daughter cell is significantly lower compared to the first cell. So an example of asymmetrical cell division, typical, for example, for stem cells. And not only that you have the imaging data, when you can see, okay, one cell is obviously smaller than the second one, but you have all the numerical data describing this process very specifically. You can also use quantitative phase imaging for the studies of cell death. So there are several papers already published that uses this technology for studies of apoptosis, necrosis, and more types of cell death. So just brief introduction, apoptosis is the programmed cell death, and necrosis is based on inflammation, for example. It's the situation when the cell ruptures and spills the content out. So here we are monitoring these two cells next to each other, and I can already tell you that the cell number 16, the one on the right, will die via apoptosis, because I can see it from the blue lines that describe its fate. So for apoptosis, there is a typical condensation of the chromatin, shrinkage of the cell area, and then the appearance of the apoptic bodies. And on the left, cell number 24, that is described by the yellow lines, dies via necrosis. So you can see that it will not decrease in the area, but there will be sudden drop in the cell dry mass. In the upper graph, that's the exact time point where the cell ruptures and the content is spelled out. And when you have the time graphs for every cell in the population, you can easily distinguish these two types of the cell death, and you can also set the exact moments when the cell death happened. So you can study, for example, induction of the cell death by different drugs. And we are quite proud that researchers here at the Masaryk University in the Czech Republic are using Q-Phase for exactly this research, and they're also combining this with the machine learning, so they will be able to recognize different types of the cell death automatically. Quantitative phase imaging can be also used for the drug testing, and the really big advantage here is that the effect that you are measuring comes only from the drug, from the treatment. The cells are not influenced by the fluorescence staining, by frequent phototoxicity. They are monitored label-free in a cell-friendly technique, and this provides artifact-free results. Just an example of another study that was conducted on our microscope, where they were monitoring different morphological parameters, and the cell dry mass is one of them, and study the effect of different treatments. Sometimes to enhance the drug uptake into the cells, the nanoparticles and especially nanocarriers that are connected with antibodies are used. So this is the example of such a research. So you can see the overlap of quantitative phase imaging and fluorescence data on the video, where from the QPI data you can monitor the cell viability, the changes of the morphology, different types of the cell death. And from the fluorescence data, you can quantify the uptake of the drug. So higher the fluorescence, higher the doxorubicin uptake here into prostate cancer cells. And another example of combination QPI and fluorescence data is this video, which shows the multimodality of our microscope. Even though it seems that the cells are blinking in green, actually this is just different acquisition intervals for the QPI and fluorescent module or camera. So here we are monitoring cells that were infected with virus, and the virus carries the GFP tag. So the cells that give positive green signal are the ones that were successfully infected with the virus. So by combining these two modalities, we can see or monitor things which are, of course, below our resolution, like drugs, antibodies, viruses, and even the small molecules and molecular changes can be monitored combining these two modalities. Another example of Q-phase application is the monitoring of cell interactions. So we can perform really long-term experiments when we can track the cells. So this is the example how cancer cells can deal with the oxidative stress by a process called engulfing. It's a type of cannibalism where one cell actually crawls into the other one and can survive the unfavored conditions there. So this is the mechanism of resistance in cancer cells. Monitoring cell interactions means also that you can monitor different cell populations in co-cultures. So this is the example of T cell, the immune cell that interacts with the cancer cell. And you can see how it affects the morphology of cancer cell. And if I skip the video a bit further, then you can see that after the interaction, cancer cell really dies, stops moving. Nice example of the work of our immune system. Yeah, now the cell died. And Aneta promised you that today there will be one application spotlight, so we decided to shed a little bit of light on the mechanobiology research and how quantitative phase imaging can be applied in those studies. So we will be talking a lot about perfusion studies, and I really like how a big company makes these nice drawing of cells, how they react to the flow system. But first of all, a little bit of physics and theory. So shear stress. Shear stress can be described as the force applied parallel to the area. Is expressed in the units of dynes per centimeter square. And this is the force that is usually experienced by the cells that are used to live in the fluid systems like vascular endothelial cells, lymphatic endothelial cells, renal endothelial cells, and others. And what is so different if the cell grows in a fluid flow and experiences these mechanical forces? So the shear stress has an important role in the cell structure, in the cell morphology, which affects, of course, the physiology and behavior of such cells. So you can see here on the image comparison of cells and their morphology, if they are cultivated under static conditions or under the fluid conditions. Oh, sorry. I think I managed to jump somewhere in the presentation. Let me just share the screen once again. I apparently jump into the presentation somewhere too far away, sorry. So the mechanical forces, the shear stress, affects many parameters in cell physiology, for example, cell adhesion, proliferation, apoptosis, expression of several genes, activity of platelets in antithrombotic cascade, and many more. So here in the introduction, I just want you to take the message that the shear stress affects significantly cell physiology and is an important force in cell biology. But how is it possible that some force can affect cell biology that significantly? It's because cells can actually sense mechanical forces and then translate this mechanical stimulus into biochemical responses. Shear stress sensing is a process how the signal is transmitted from the cell membrane into cell interior, and there are different signaling pathways how cells can do that. There's a key role of several signaling molecules, and the shear stress can be sensed, for example, by ion channels, growth factor receptors, as described in the picture, by cytoskeletal molecules, adhesion proteins, and some more. However, there are multiple pathways involved in this mechanotransduction effect, and these pathways are not fully understood yet. And we see that this could be a knowledge gap, an opportunity for quantitative phase imaging to be employed in those research areas. What research areas am I talking in particular? So, for example, vascular pathology. I mentioned that endothelial cells experience shear stress every day, and if the shear stress increases, there can be some pathological changes in their physiology. Cancer research. It's important to study the mechanical properties of cancer cells because in the tumor progression phase, the endothelial mesenchymal transformation happens in the cells. So the cells cancer cells, they have to change their mechanical properties to be able to leave the site of the primary tumor, get into the bloodstream, and then start a secondary tumor somewhere else. So they have to significantly change their mechanical properties to be able to survive all these changes of the conditions. Both of these areas are highly applicable for QPI, and secondly, these studies, when you study the effect of the flow on the cells, are often conducted in the microfluidic systems, and it has been already proven by several papers that the quantitative phase imaging can be also conducted in these microfluidic devices. So let's see some research examples, what has been already studied in these areas. So first, I would like to introduce the paper from a Chinese research team that focuses on osteocytes, because these cells are mechanosensors in bone, so they expose the cells to pulses of the shear stress, and they use quantitative phase imaging to monitor them. So first of all, they reconstructed the phase maps, similar as we do in our microscope, and then they calculated the phase difference, meaning that on the left you can see the phase map, on the left on the video, which tells you in the colors. The red color means that there is a higher phase shift, higher cell dry mass compared to the surroundings, and the phase difference is just the difference between these two images. So you can see the red color is the pixels where cell dry mass was increasing compared to the blue pixels where the cell dry mass decreased. Second paper I would like to introduce comes from Australia, from Cambridge, and it's about label-free multimodal quantitative imaging flow SL for intra-thrombus formation in vitro. So in this research, scientists were studying platelets and how they actually react to the flow, how they became adhesive and then activated, so more platelets are combined together in the process of embolization. So this research actually is based on the idea that the refractive index of platelets is stable, so you can use it as endogenous label and study the changes in the dry mass. So they were studying the spreading of these cells and the activity in the collagen coating. And also this can be used for studies of antithrombic agents, so you can test which drug affects the platelets activation and spreading and by what mechanisms. And then I've also read and prepared for today 2 papers from the North Carolina, from the Duke University. First is the measurement of the shear modulus and correlation with atomic force microscopy. So atomic force microscopy is a traditional technique for studying the mechanical properties of cells, but it's a contact technique compared to the QPI, which is of course contactless and label-free. So again, you can see the cell dry mass, not cell dry mass, phase differences on the color-coded images. And they also studied how the cells react to the exposure of cytochalasin D, which is a toxic agent that depolymerizes actin filaments, which of course affects the cell stiffness. And they compared two numbers. From QPI they derived shear modulus, and from AFM they derived the Young modulus. And in the paper they describe how these two techniques nicely correlate together. From the State Research Group comes this papers, which is really interesting, studies of the lung cells. And first they used a non-cancer human lung cells, and they exposed them to arsenic treatment for several weeks to activate it, their change into carcinogen cancer cells, because arsenic is a typical carcinogen, especially relevant for lung cells. And then they seeded the cells in agar. They measured cell stiffness, and they found out that it's a sensitive parameter for subtle shifts in the cell morphology, and also it can serve as a sensitive biomarker for the cancer transformation progression. So I think even just 4, but these research examples show that QPI is a powerful tool and non-contact and label-free tool for exploring cell mechanics. And we are really proud that also on the Q-phase microscope we have already 2 papers published, not we, of course, our users, where they used Q-phase, they combined it with the flow systems, and they studied cell stiffness. So the first paper described the changes of the cell stiffness after the treatment of 3 anti-cancer drugs on prostate cancer cells. And the theory behind is that cancer cells within tumors are typically much softer than the cells, the surrounding tissue. But when cells decide to create metastasis, they are stiffer compared to the primary tumor cells. And they were studying the effect of 3 drugs on exactly this stiffness. And again, they correlated the QPI technology with AFM and confirmed that these technologies correlate nicely in terms of measuring cell stiffness, cell structure, and cell functions. The second paper from the same research group describes the viscoelasticity measurement, and it's more about the physics and math behind. But again, confirms that QPR is the non-invasive method for cell viscoelasticity measurement. The images just show how the fluid system is employed in our microscope, and the image on the left shows the changes of the phase shift in radiance after pulses of the fluid are sent to the system, and cells react to those pulses of different force. So it seems that I was speaking quite fast. I hope you understand also the spotlight part, and let me just summarize once again the main advantages of quantitative phase imaging technology. So first of all, it's labor-free technique, meaning that it's non-invasive, non-disturbing. The cells really like this technology, and there is no toxicity coming from the label because there is no need to use labels for visualizing the cells. It provides quantitative data, directly the data on cell dry mass, but also, especially in these flow studies, cell dry mass distribution across the time, they allow to precisely measure the refractive index. This technology is of low phototoxicity because it doesn't depend on the cell intensity, only on the interference of the 2 light beams. So it's suitable for live cell imaging. There's no photobleaching because there is no need of using fluorescent dyes. So combining with the stage-up incubator, it's really suitable for long-term observation. It's a fast acquisition technology, providing high contrast data of high quality. And if it's combined with the other imaging technologies like fluorescent mode, it allows to combine these 2 modalities and use, for example, fluorescent imaging to study the specific pathways. In case of the mechanical studies, you can study specific mechanotransduction pathways to explore this research. I think that's all from my side.